Bismillah ar-Rahman rahim. This is Hafiz Muzammil Rahman with our new tutorial. In today's tutorial, we will try to learn about the protein docking. In our previous tutorial, we have talked about the protein ligand docking by using the Autodock Vina or Autodock tool. The two tutorial are already available on our YouTube channel. Uh, First one is about the multiple ligand docking, that is about Autodock Vina, and other one is about Autodock tool. In today's tutorial, we will try to learn about how we can perform docking of the protein, uh, or if we are interested in docking to protein or DNA, or protein or peptide, or even if we are going to dock protein with a ligand by using an online tool. So in today's tutorial, we will try to learn how we can use the online tools to perform the docking. If you are not easy with the manual docking, and if you don't have the expertise in Autodock or Vina, so there is no need to worry now. You can do an automated or online docking. So first of all, we will talk about the PatchDock, and then we will talk about the ClusPro, and in the end we will try to analyze the result. So first of all, go to the Google and type PatchDock. Then this page will be open, and then click PatchDock server. It will sometimes show I'm not a robot. You will have to click it and then go to the server. PatchDock, as it is written over here, this is the molecular docking algorithm that is based on the shape complementarity principle. It will match the shapes of the molecule, and accordingly it will dock these two molecule or two receptors. If you will click on the help or about the PatchDock, yes, if you will click on this, so the PatchDock algorithm is for molecular docking. And if you have any type of molecule that may be a protein, the DNA, the peptide, or drug, this PatchDock is equally suitable for all these type of docking. So the output will be a list of potential complexes that will be sorted by the shape complementarity criteria. There are three things which on which this server work. The first is the molecular shape representation. It in this module or in this criteria, it recognize the shape of the molecule, and then recognize the and then detect the segmentation or the geometric patches, like how the surface is oriented, whether it is the convex patch or concave patch, what is the shape. Like you can imagine the jigsaw puzzle in which you match the shape, and then accordingly you fit between the two particle or the two shapes of any items. So in this way, it will firstly analyze the shape, the geometric patches, and then these patches will be filtered, and then these patches will be there will be another criteria in which it will be decided whether these patches are good or not. So these will be filtered accordingly, and only patch with the host spot residues are retained, in which there is some interaction between the two receptors or two molecules. The second one is the criteria for which it on which it work, the PatchDock, that is the surface patch matching. In this criteria, it make the cluster post cluster matching technique, and the patch that was detected in the previous step, that is the concave patch, are matched with the convex, and flat patches with any type of the patches, so these patches will be matched. So this is a hybrid of geometric hatching and post clustering matching. In this step there are three steps, as I have already discussed. The first one is the molecular shape representation, in which there will be the analysis of the shape, analysis of the patches, whether they are convex, whether they are concave, whether they are flat, and then they will be recognized. And in this the second step is the surface patch matching, in which these will be these patches which are detected in the previous step, they will be more analyzed and more filtered. And the third one is the filtering and scoring, and in this the candidate complexes or the filtered patches in the previous sections, they will be examined, and they will be discard all the complexes with the unacceptable penetration of the atom of the receptor to the atom of the ligand, and those which will have good geometric shape complementarity score, they will be ranked accordingly and they will be retained accordingly. So in this way this patch doc work. So you can click over here to help, and all the information about the receptor molecule, ligand, and clustering RMSD, as I have talked about it. So all these things, they are available on the help. So we are clicking on the web server again. So this page will be open. So there are some many options. The first one is the receptor molecule. Here you can type the PDB ID, or here you can choose the file. If you have already downloaded the PDB file, you can choose, or you can drop over here. In this, the ligand molecule, you can also the type of the PDB, and your both receptor and the ligand, they should be in the PDB format. So you can download the receptor and the ligand in the PDB format, or you can upload your PDB file if you have already available that. There are some examples, like if you have a PDB molecule, and it has many chain, so you can type the name, like they have given 2KAI. So the then colon, the chain of this molecule has many chains, so we are interested in chain A. So in this way you can select the chain as well. And if you have a PDB molecule and it is many chain, so you can tell the server on which chain you want to dock your receptor, sorry, ligand, and accordingly same with the ligand. So if you have downloaded the PDB file already, so you can upload the file over here. We are going to PDB first, and I am going to dock 1A22. This is human growth hormone and bound to a single receptor. One is the hormone and other is the receptor. B, chain A is the hormone and chain B is the receptor. So these are the two chains. In today's tutorial, yes, this one. So we will work on the receptor and the growth hormone, but we are not going to follow completely this PDB. In our one of our research was on this type of growth hormone and the receptor. So we have choose the growth hormone, and we have choose the receptor of the Babulus. That is the domestic water buffalo. We have choose the growth hormone of this buffalo and the receptor. Because the growth hormone of the buffalo and the human, they are somewhat similar to each other. So we have chosen them to dock with each other. So I have already prepared the file. This is the file of the receptor. So I will drag it over here. You can drag it or you can just click the choose file as well. Okay, so this is drag over here, and this is the receptor, and the ligand, the second, this is the receptor, growth hormone receptor. So I want to dock the growth hormone receptor with the growth hormone. So this is the growth hormone, Bibulus GH, the growth hormone. I have prepared the file. You can directly download the protein molecules or the files, and you can directly upload it. There is no need to prepare the file, but I would recommend to prepare the uh, molecule before going to dock. But you can go directly as well. How you will be able to prepare the protein or ligand? You can consider my previous tutorial on the protein docking, in which I have discussed how we will be able to prepare our proteins. Uh, so the protein and the receptor and the ligand, both are the protein. They are uploaded, and here you will have to give your email ID. I have given the email ID of mine, that is muzammil.phd.ibb@pu.edu.pk. So this is my email ID. Uh, the clustering RMSD, that is 4. This is the default. This is okay. Don't change. There is no need to change. In this complex type, you can keep it default as well, but if you are interested in any specific type of docking, like if you have a enzyme and inhibitor, so you can click on enzyme and inhibitor. If you have antibody antigen docking, so you can click over here to antigen antibody. And if you have a small molecule or drug, with you want to dock with that you want to dock the drug with the protein, so you can choose this option as well. I'll go with the default. There is some advanced option. If you will click on show, so the default option will be here. There is the receptor binding site. If you do not know about the receptor binding site, you can predict the receptor binding site as well. For this, you can use this BS spread server. You can just type BS spread on the Google, so this page will be open. You can paste the sequence because it work on the sequence of the amino acid. It is the basically neural network based algorithm for predicting the binding site. So you will have to paste the sequence in the FASTA format, or you can upload the file. And email is mandatory. You must have an institutional email ID. Another one is CastP. CastP is a very famous server. If you will upload your protein over here, so it will predict the protein binding pocket like this one in this case. So you can upload your own PDB file. So accordingly, you will be able to analyze the binding sites of your proteins. So here you can choose like this. There are some other options. In this way, I will make another separate tutorial on the how you will be able to find the protein binding site if you do not know, and if they are not reported already. So how you will be able to predict the binding site? If you have a native protein and you have modeled it by yourself, then how you will be able to find out the protein binding site? I'll make a tutorial, separate tutorial on this. But anyway, you can upload your file over here, and accordingly you will be able to know which binding pocket is suitable for binding. Another server is, that is Coach, protein ligand binding site. You This is a very important and very good server. So you can upload your protein over here, and accordingly you can, yes, accordingly there is again email ID mandatory. You will have to give your email ID of your institution email ID. And before this you will have to register yourself. So it is preferred use your institutional email ID rather than any other email ID. Okay, so these three server, they can be used for the protein binding site if you do not know the protein binding site. And if you do not specify the binding site of the protein, so there is no need to worry about it. You can keep as it, you there is no need to choose anything in them. The server will automatically dock your receptor with your ligand, so there is no need to worry again. So I will click here on submit. That is submitted. It will take a while. Yes, uh, the job has been submitted and the result will be sent to this email ID, which I have given this PatchDock server. And then there will be they will send an email with this link. So this is the link. You will that is the PatchDock results. So when you will click on it, so it will give you 20 solutions. Okay, so this is the score. The more is the score, the better is the docking. This is the surface area, and these are the transformation, or these are the different orientations. So these are the different results. The default, by default it showed 20, and you can go further as well. If you want to download the 10 complexes, so you can click on this go, or you can click the solution table and transformation file. These all will be downloaded. So if you will click the transformation file, it will show this transformation file. You can copy this transformation file, or if you click to solution table, so it will give you the solution table. But there is another option which is very good, and many tutorial on the PatchDock they didn't use this option, which is very important, available to PatchDock, and that is you can refine these docking solutions by using the FireDock. This is again a very good server. So the refined best solution with the FireDock, you can use your 10 best solution to refine them through the FireDock by the option clicking the option go. It will, the top 10 were transferred to FireDock for the refinement, and accordingly the link will be sent to your email address which you have given to the PatchDock. So this is the FireDock results, and if you will click on it, so these are the results. In this there are different type of energies are available. So if you only use the result of the PatchDock, which was like this, so there is only the score. you do not know about the energies and other things. And if you refine your docking solutions with the FireDock, so you will be able to get result in this way. So these are the global energy, that is minus 20, 22.46, the attractive van der Waal forces and the repulsive van der Waal. So these are all available if you will refine your solution with the FireDock. Okay, so accordingly you can download, you can click here on the download the best structures, the folder will be downloaded. So if you will download the folder, this is the folder, and then these 10 solution will be available in the FireDock solution folder. So you can go for analysis as well. Another server is the ClusPro. You can type on the Google ClusPro. This page will be open of the Boston University. So you will have to make your account on this ClusPro. ClusPro is the very cited server. PatchDock is the most cited server as well, and the ClusPro is the also very best server for the protein docking, protein peptide docking. Even they offer more as well. So if you will log in over here, so there are different type of docking as well. You can type you can dock the RNA, you can dock the DNA, you can dock the small peptide as well. So these are the peptide docking option. Here you can do peptide. These are dock option. Even it has a very important feature in the ClusPro, that is the dimer classification. If you have a chain which is present in the form of dimer, and many receptors, transmembrane receptor, they're usually present in the form of the dimer. So you can use this dimer classification for to dock such type of receptor as well. So the ClusPro is a very good server as well. So you can go to the dock. You can give the job name any, like I'm giving the job name dock or docking. So you can use the CPU or GPU for your docking process, and similarly, as you have uploaded the protein or receptor file onto the PatchDock, similarly you can type the PDB ID, or you can click the upload file, then here upload file as well, and accordingly you can track. Like this is the ligand, REC, this is the short form of receptor. I usually write REC in the short form. So this is the receptor and this is the ligand. And if you have a multi-chain molecule, so you can specify the chain as well, at which chain you want to dock your ligand. So if it has, let's suppose it has a chain A, B, C, and D, and I want to dock my ligand with the chain C, so I can type here chain C, and if ligand has multiple chain, so and the chain D is important, so we can type over here the chain name accordingly. Okay, so there are also advanced options. You can click over here for the attraction and repulsive residues as well. If you know these residues, they can be find out by the literature, and if you know these residues, you can type, or you can give your option over here. There is structure modification, and if you want to remove something in like if you have downloaded the PDB, or if you the structure and you have uploaded as it, and it has already bound ligand and other things, so you can remove accordingly. So this is multimer docking beta version, uses the upload receptor as all unit of the multimer, as I have already discussed this. The other mode, the antibody modes, if you want the antibody as receptor, antigen as ligand, so you can do the docking, you can use this as well. You know, this is very important for docking. If you have antibody and antigen, docking as there are many tutorial on the vaccine for the COVID-19. So this is the best to dock the antigen and antibody interactions, to check out antibody antigen interactions. uh, these are the uh, heparin ligand. This be used for the heparin ligand if you have such type of ligand, and these are the other options. You can read it accordingly. If you want to give some restraints, they are already available, the option already available like that. So they are already checked, so keep all these things as default. And if you have antibody antigen docking, so you can click here and you can check like this. Okay, so after this you can click on the dock. When you will dock, this page will be shown, the queue. So your name, your job ID and the name and the username, that will be shown over here, and the status will be shown over here, copying to local computer. Or if okay, I'm just going to do it for you again. This is the receptor. I have already docked it, but I'm doing it again for you, and how you can monitor what is happening to your job. This is the ligand. Okay, so by clicking, sorry, job name is, oh my god, I have clicked without the job name. Let's wait. What is going to, I'm just going to cancel it, and then job name is again doc. Okay, so I am choosing the GPU, so then clicked the dock. So now it will be submitted on the supercomputer, and then accordingly you will be able to monitor your job. If it is like this, you will have to wait for some time. Why it gets slow? Okay, no worry. uh, you will be you will receive an email uh, regarding the completion of your job, and you can directly monitor by clicking the queue or the results. And if you're like this, what is the queue? If you will click the queue, it will show you whether your job has been submitted or not. Okay, so these are many tabs of the ClusPro. So these were the models, or I have already docked the results. You can, yes, this is the REC receptor, BB, babulous growth hormone. So if you will, or this is the REC human growth hormone. I have already done the docking. They will keep your job for the 2 month, and after the 2 month, the results will be automatically deleted. For the 2 month, your result will be present on the server as well. And if you will click on the job name, so by default it will display the 10 models. These are the 10 models that by default it displays. You can click here to show more. So now it will show you 27 model. It start from the 0 0 1, so it is showing 26, or the total is 27 model. You can click here to watch the model score. So it has a default criteria, that is the repulsion, attraction, electrostatic forces. On these type of forces, it predict the scores. This is the center, because the ClusPro is working like the clustering. It make the cluster, and then accordingly it will choose the cluster with the lowest energy. So that is the center. It means how the ClusPro work. You can go to the help, and then there is a question over here, how the ClusPro work. Yes, so here is the answer. We rotate the ligand with the 70,000 rotation, and for each rotation we translate the ligand in XYZ relative to the receptor on a rigid. They make the receptor rigid, and then they check the orientation or rotation of the ligand, and then accordingly the best rotation and will be used, or out of the 70,000 rotation we choose the 1,000 rotation or translation combination that have the lowest score. So in this way they choose the cluster. So we do a greedy clustering of these 1,000 ligand positions. They choose the 1,000 ligand different position within 9 angstrom with the alpha carbon RMSD radius, and then accordingly it show the results. So in this way this work. It is very important server. You can read all these things by going to help, and these are the scoring. So by default people choose the zero, the first zero cluster, the rank 0, and with this the highest score and the lowest energy. But I will recommend you should look the complexes manually, and then you will be able which complex is good, and the binding pose of the complex is very nearer to the experimental results. So I have downloaded it, the complexes from the PatchDock as well, and then from the ClusPro as well. So this was the my original PDB with the human growth hormone and the receptor. And what we have done, we have docked the the growth hormone receptor with the growth hormone of the baboonus. So you can check whether your dock pose is good or not, or you can validate it. How you will be able to validate it? First of all, let me change the background for you. This is the background. Now I'm going to white it for you. Okay, so I have changed the background. This is the Chimera tool. I have already discussed the Chimera in my previous tutorial, how you will be able to download the Chimera. And if you do not know about it, so I am going to do it again for you. Go to the Google, write the Chimera tool. This page will be open. No, this is not Chimera, University of California, San Francisco. Yes, this is the Chimera. This is by the University of California. You can click the download over here, and accordingly, if you have a system of the Microsoft Windows, if you have Mac, if you have Linux, you can accordingly download the version according to your platform. So I have Mac, so I have downloaded this one, and therefore it is in the magenta. It is already downloaded. Okay, so how you will be able to make a good picture, I will make another separate tutorial, but you can go to the action and then color and then all options. Action, color, and all options. And all option, there is here. This is the background. And when you will click the background, so click here on white, so background will be white. Okay. So this is the PDB which is already present into the PDB protein data bank. So I want to check whether my results are okay or not, or my results are good or not. So I want to validate. If you have a protein and small ligand, the same in the same way you can validate your docking as well. So this is my receptor and the babulous growth hormone chain. So I have uploaded both of them. Yes, you can now see both has been uploaded. And the one thing is very important, the receptor of the babulous, that is similar to human. So we have predicted the structure of the babulous of this by ourselves. So this was the our project. This was back in 2012 or 14. We have uploaded the sequences in the NCBA as well. So this, okay, so now we want to check whether our docking is good or not. So by uploading both of the complexes, how I have uploaded, first I have uploaded the complexes simply. You can upload the complex by clicking the open and select accordingly, and then here there is the blue window. If you will click on over here, so accordingly you can select another complex as well, or if, again you can go to the file and open and select the other one as well. If you are interested, if you do not know how I have done it, I will do it again for you. This is the summary of the already uploaded file. So this is the file which I have downloaded from the PDB. Okay, so I'm go for the option and color and then all option. Here is the background. Click on the white, so background will be white. Okay, now here at the right side bottom, this option is clicked and then the receptor has been selected, or you can manually you can do it manually by going to the file and then open. Okay, so these both complexes has been uploaded into the interface of the Chimera. Now I want to check it. So go to the tool. There is the option structure comparison, and in structure comparison this is the Matchmaker. So click on the Matchmaker. This window will be uploaded. Select the reference model, the model which you have downloaded from the PDB, and then the structure on which you want to match. Okay, so rest the default option, keep it like the default, and then just click on to the OK. So this is the RMSTs 2, and okay, I am going to make it more easily visible. So go to the action and click on the focus. So now you can look, this magenta or the cyano color is our docked complex, and in this brownish color, that is the PDB which we have downloaded from the PDB bank. So you can look, this is the growth hormone in the helices, and the hormone which is present in the cyano, this is from the buffalo, and the hormone that is into the brownish color. This is of the human, and they are similar, almost similar to each other. So therefore uh, we have docked it with the receptor as well. So now you can see the position of both of the hormone and the receptor. They are aligned with each other, they are superimposed with each other, and the position is little bit changed, and it show the orientation or the pose where you have docked your receptor, that your ligand with the receptor, that is good. So in this way you can validate your results whether they are good or not. So if you want to select or you want to delete any chain, so you can go to the chain A, and if you want to delete the receptor of the buffalo, so that has been deleted. Okay, so in this way you can do all these things. In the last, if you want to make a very good picture, so I have already uploaded a tutorial onto generation of good diagrams, but here I will show you little bit. I will do it fastly for you. Just go to the select, then chain, then we have selected chain A, then go to the action and color. I have selected the purple color. Okay, then again go to the selection and clear selection. Okay, so this, the color has been changed, and this is in the pink color. Again go to the chain, select B, and okay, so I am going to make it blue. Okay, then again clear selections, again select chain A, action and surface, show. So this will be in the form of, then this will be shown in the surface. Okay, so you can change the transparency of the surface as well. If you want to 40% transparent, we will change it accordingly. Okay, so I'm going to again chain B, and then select chain B, and then action and surface and show. So this is the surface, and again action, surface, and transparency is 40%. So here click clear selection. So in this way you have made it like this. So okay, so this shown a very good surface, and if you change the color or the transparency as well, so you can do like this. Okay, so if you want to very beautiful, so you can change it accordingly according to your need. And the last thing, you want to save the image, the publishable image, click onto the file and then save image. Okay, do nothing, just here at the print resolution DPI just type 300. All the other things keep them default. Just type here, if because I usually work on the 300, so it is selected default, and if there is something some any values, so you can change it and make it 300, or you can make it 600 as well. So I will make it 300, and then pro PPI, I am going to the name to this PPI, the image name is PPI, and selected directory is desktop. So okay, click on the save, and then you will know how much in the quality of the picture. So it has start working. It will take some time because the quality will be very high. Okay, so now we are going to check the interaction in the 2D form as well. So I'm going to do it quickly for you. Okay, so you can go to the PDB sum. Okay, and then PDB sum, here you can click on the generate, and you can upload your file over here. And by uploading the file over here, you will have to type your email address and then upload it accordingly. Then such page will open. The page which will be open, it will be like that. So this is your, you will receive an email, okay, like this. This is the PDB sum custom generated page, and then this is the direct link. When you will click, you will go to the page directly. Okay, so yes, this is the receptor, and this is the growth hormone. Okay, so you will have to click on this protein. So this is chain A, this is chain B, and there are interactions. The red line shows salt bridges, the yellow line shows disulfide bonds, and the blue hydrogen bonds, and this orange non-bonded interactions, non-bonded contacts. So these are the results. 6 salt bridges, hydrogen bond is 11, and number of non-bonded contacts are 155. So in this way, this is the result. So if you will click on the list of interaction here, so these list of interaction will be shown in this way. So you can copy and paste into the text editor or your Word file. So this is the number of non-bonding contacts, number of hydrogen bond, number of salt bridges, with all the details. Okay, last thing is, if you will click on the the PDF format, so this picture will be downloaded in the PDF format, so you can use it for the publication. So this was all about the 2D interactions. Okay, whether yes, it has completed. So I'm just going to the desktop and want to check whether yes, this is our picture of the 3D picture. Okay, it has opened. You can check, you can zoom it and just check the quality. There is no problem of the pixel. Very clear results. Oh my god, yes, thanks to the Chimera. Very good results. So you can publish your structure like that. You can change the color, you can change the surface transparency accordingly. You can practice it, and then you will be able to save the picture in a very high good quality. This is all about the today's tutorial. That was the protein docking. For the next tutorial, we will try to talk on the more topics, and if you think it is good and I am going in a good way to promote the free education, so just subscribe the channel and comment, like, and share to other. Bye bye, take care.